Good morning, my dear students. Today I am talk about gene transfer methods in plants. There are two different types of gene transfer methods used at present. One is vector mediated gene transfer methods, and another one is direct or vectorless DNA transfer methods. The vector mediated gene transfer methods example is Agrobacterium mediated gene transfer method. And the second method is the direct or vectorless DNA transfer methods. There are two different types of methods present in this method. One is physical method, another one is chemical method. The physical method, electroporation, microprojectile, particle bombardment, microinjection, liposome fusion, silicon carbide fibers. These are the methods are used in the physical methods. And chemical methods, polyethylene glycol mediated, and then diethyl aminoethyl dextran mediated gene transfer method. These are the methods at present widely used for transferring the genetic material into the plant genome. Okay, now we are going to see in detail about agrobacterium mediated gene transfer methods. Now, agrobacterium mediated gene transfer method. In this method, agrobacterium is the gram-negative bacterium. It is a rod-shaped, motile bacterium, and this bacterium belongs to the family of Rhizobiaceae. Okay, look the pictures. The agrobacterium usually present in the soil, and then the plants are secreting some chemical components. From these chemical components attract the agrobacterium. These two combinations are reason for the transferring the genetic material into the plant genome. It happens in naturally, so it is called nature's most effective plant genetic engineering methods. Some workers consider this bacterium as the natural expert of inter-kingdom gene transfer. Okay, in this bacterium, there are two different types of bacteriums are used naturally. One is Agrobacterium tumefaciens that induces the ground gall diseases, and then Agrobacterium rhizogens that induces hairy root diseases. Okay, uh, the in this agrobacterium, the special plasmid regions are presented. The plasmid DNA, in this plasmid are called Ti plasmid. This Ti plasmid is the reason for the transferring the genetic material into the plant genome. Look, in this picture, the Ti plasmids are present in detail. Okay, in this Ti plasmids are having tDNA region, and then virulence region, and then replicator region, and then opine catabolisms region. Okay, the tDNA region, the region is a special regions. In these regions are reason for transferring the genetic material into the plant genome. Okay, and then virulence regions also present in it. These genes are responsible for transfer of the tDNA into the host plant or located outside the tDNA, and this region referred to as virulence region. The virulence regions codes for proteins involved in tDNA transfer. At least nine virulent genes are present in it. There are virulence A, virulence G, virulence B1, virulence C1, virulence D1, D2, and D4, and virulence E1 and E2. These are the virulence regions are responsible for transferring the tDNA into the plant genome, and then opine catabolism region. This region codes for proteins involved in uptake and metabolisms of proteins, and also origin of replication regions also presented, that is responsible for the origin of DNA replication, which permits the Ti plasmid to be stably maintained in Agrobacterium tumefaciens. Okay, and then how the tDNA regions are transferred and get integration into the plants. Look, this is the pictures, diagrammatic pictures, representations of tDNA transfer, and it is integrations into host plant cell genome. Okay, the tDNA transfer having so different steps are present in it. One is first one is signal induction to agrobacterium, and the second one is attachment of agrobacterium to plant cells, and the third one is production of virulence proteins, and the fourth one is production of tDNA strand, and the fifth one is transfer of tDNA out of agrobacterium, and sixth one is transfer of tDNA into plant cells and integration. The first one, the signal induction to agrobacterium. In this method, the wounded plant cells release a certain chemical. These are the phenolic compounds and the sugars which are recognized as signals by agrobacterium. Okay, the chemicals are secreted from the plants, and then the agrobacteria move towards uh, the plant cells. The signal induced result in the sequence of biochemical events in agrobacterium that ultimately helps in the transfer of tDNA of T plasmid. And then second one is, and then after the attraction takes place, then the agrobacterium's attachment to the plant cells. Okay, attachments. Then production of virulence protein. After attachment takes place, the virulence proteins, as the signal induction occurs in the agrobacterium cell attached to the plant cell, a series of event takes place that result in the production of virulence proteins. To start with, the signal induction by phenolics. Phenolic stimulates by virulence A, which in turn activates the virulence G. This induces expression of virulent genes of Ti plasmid to produce the corresponding virulence proteins D1, D2, E2, and B, etc. Okay, and then production of tDNA strand. Then the tDNA strands or comes out from the Ti plasmid, and then the virulence genes are protectively transferring the tDNA into the plant genome. The virulence proteins, look, so many virulent genes are responsible for encoding the tDNA, and then without any damages takes place by with the help of the virulence genes to protectively the tDNA strands get enter into the plant genome, and then the tDNA regions are and get enter into the nuclear pore complex, and then it is transferred into the plant genome, and then transcription, translation, the production of auxin, cytokinin, and opine. These are the actions are takes place in the plant genome, and then the selected DNA is transferred into the plant cells, and then the plants are getting the foreign DNA characteristic features into the plants. These are the reason for the production of genetically modified plants BT cotton, BT brinjal, and then these are the reason for production of this kind of plants in future. Okay, thank you very much.